Hi learners, you're welcome to our 7th lesson in integrated science. Hope you are doing well. In our previous lesson under diffusion and osmosis, we touched on diffusion, the definition of diffusion. We also looked at 2 demonstrations of how diffusion takes place in air and in liquid. And then we proceeded to look at examples of diffusion in plants and animals. Today, you are expected by the end of the lesson to be able to explain the term osmosis and to demonstrate osmosis in non-living tissue. 1. Explain the term osmosis. 2. Demonstrate osmosis in non-living tissue. So, what is osmosis? We are saying that osmosis is the movement of water molecules from a region of lower molecular concentration to a region of higher molecular concentration through a semi-permeable membrane. So in the definition we have keywords: movement of water molecules from a region of lower molecular concentration. That's in a region where any other molecules exist only in small amount. That's why we say lower molecular concentration. Therefore, in this region, water is so much here. To a region of higher molecular concentration, this region has so much of solute particles but less water, and the movement is across a semi-permeable membrane. Now, a permeable membrane is a membrane that allows substances to pass through. A semi-permeable membrane will be a membrane that allows only certain substances to pass through. And in this case, it allows only water molecules to pass through, but do not allow any other solid particles to pass through. Now, what is the second definition? Another way of defining osmosis. Good. Is the movement of solvent molecules from a dilute solution to a concentrated solution across a semi-permeable membrane. So in this also, there's a movement of solvent molecules, usually water, from a dilute solution, a solution with much water, that's dilute solution, or weaker solution, to a concentrated solution, that's a solution with so much solute particles like salt or sugar across a semi-permeable membrane. So you can define osmosis by one of these definitions. Now let's move on to the diagrammatic illustration of osmosis. The definition, how is it represented diagrammatically? So looking at the diagram carefully, we have in this beaker a semi-permeable membrane separating a concentrated solution of sugar molecules from water molecules. So this is the water molecules and this is the sugar molecules. Now look at the movement. As we said, the movement is from dilute solution to concentrated solution, or from where water is so much to where water is less. So the arrow indicates the movement. And the movement is through a semi-permeable membrane, as you can see. A semi-permeable membrane. Okay? So the result, after some time, 6 or 7 hours, we have water moving so much into this region. Therefore, the level of solution here becomes higher and that of water will decrease. So this represents osmosis, pictorially or diagrammatically. Now let's move on to look at the experiments. But before then, let's study factors that affect the rate of osmosis. Factors that affect the rate of osmosis, or what should be the condition that will cause water molecules to move from one region to a region where there is high molecular concentration. So, one, there should be a concentration gradient. Concentration gradient. We explained concentration gradient under diffusion as the difference between two concentrations. So in this case, if the concentration or the difference between the concentrations of the two regions is very big or large, there's a huge difference, then we are saying that water molecules will easily move to the other region. But if the difference is not that big, then it's going to be difficult, or it's going to be very slow when it comes to movement of water from one region to the other. So one factor is concentration gradient. The next is there should be presence of semi-permeable membrane. There should be presence of semi-permeable membrane. If there is no semi-permeable membrane, water will not move from one region to the other. Lastly, temperature. The higher the temperature, the higher or the faster the molecules of water will move to the next region. And the lower the temperature, the lower the rate at which water molecules moves to the next region. So these are the factors that affect the rate of osmosis. 1, there should be concentration gradient. 2, presence of semi-permeable membrane. And 3, there should be temperature. Now, hope you are following the lesson. Now, let's move on to look at experiment to demonstrate osmosis in non-living tissue. So the experiment title is experiment to demonstrate osmosis in non-living tissue. Good. So what is the aim? The aim is to demonstrate osmosis in a non-living tissue. That's the aim, to demonstrate osmosis in a non-living tissue. Now, what are the materials we need, or the tools and apparatus? We need retort stand and clamp. We need a thistle funnel, which you soon see. We need a parchment paper or the baking paper. And then we need concentrated sugar solution. We need distilled water. And we need beaker. So let's look at the diagram. Good. The diagram will explain everything in detail. Now, this is the beaker. This is a thistle funnel. This is a thistle funnel with a long tube, turned upside down. Now, surrounding the thistle funnel is the parchment paper. Is a parchment paper. Now, the parchment paper serves as a semi-permeable membrane. The parchment paper serves as a semi-permeable membrane. Good. And around the thistle funnel in the beaker is a solvent, more of water. That is water, okay? Now, in the second diagram shows that the thistle funnel is filled with the concentrated sugar solution. So you can see we are filling the tube with concentrated sugar solution. So the question is, what will happen at the end of the experiment? So let's look at the method. So this is a diagram. Now let's look at the statement describing the diagram. So under procedure 1, we are saying that a piece of parchment paper is fitted over the mouth of a thistle funnel, as we've seen. 2, the funnel is inverted, that is turned upside down, so that the covered mouth is at the bottom, and then it is filled with concentrated sugar or salt solution. The level of the sugar solution is marked with ink. So we will now mark the level of the sugar solution with ink, so that if there's any movement of solution, we can easily detect. Then third, a beaker is filled with water and the distal funnel is immersed or put into the beaker containing the water, as you've seen in the diagram. Then finally, we will clamp the tube of the distal funnel in a vertical position. Now we will now allow the setup to stand for 6 hours so that we can see any visible changes. Now let's check out the observation. The observation, according to osmosis, we will observe that the level of the sugar solution in the distal funnel will rise. The water in the beaker will reduce. So that's what we will observe after 6 hours. Now, what is the conclusion? The conclusion of the matter: the water molecule in the beaker is high concentration of water molecules. Take note, the water molecule in the beaker is high concentration of water molecules. Water is so much in the beaker. The water will now move from the beaker into the distal funnel with the parchment paper serving as a semi-permeable membrane. So it's clear the parchment paper serving as a semi-permeable membrane will not allow the water to move from the beaker into the distal funnel. This explains why the water level in the distal funnel rose and the water in the beaker reduced. So this is a diagram summary. After 6 hours, this is what we'll see. The concentrated sugar solution here, which was here, this is it, begin to be diluted as water moves into the tube and it rises. So this is the former level, this is a new level. All right. Hope you have understood the lesson in its fullest. Now, we began by looking at the definition of osmosis, and we studied the factors that affect the rates of osmosis. Then finally, we touched on osmosis in non-living tissue. That is the summary work. All right. For our advanced learners, today we did osmosis in non-living tissue. So try to read on osmosis in a living tissue. Okay? Congratulations for coming this far with me. This is all time will have us. Try and do the exercise, upload it onto the page for me to access, and the results will be given to you by the close of this weekend. Till we meet again. Bye-bye.